Assalamu alaikum. This is Zeinab Mazloum and I will be explaining Document 8 of Chapter 7, Diagnostic Applications of Antibody Properties. By now, you all know that antibodies are proteins produced by plasma cells during the immune response against bacteria to mediate their elimination by different components of the immune system, toxins to neutralize them and prevent their action on body cells, and viruses to neutralize them and prevent them from infecting their target cell. So almost every intruder induces the formation and secretion of specific antibodies. Now, it is true that your body already contains a pool of billions of B lymphocytes that can recognize any antigen present in nature by their membrane-bound antibodies, but the secretion of antibodies is only acquired upon the infection with an antigen. That's why when a specific antibody against an antigen is detected in your blood, urine, or any of your tissues, then you must be infected with this particular antigen. So for this document, we will discuss how antibodies are used as biological tools to detect any infection in the following lab techniques: agglutination reaction, immunodiffusion in gel, ELISA, and immunofluorescence. So notice that this document doesn't discuss the evolution of any immune response in the body. It only discusses lab techniques that use antibodies to detect infections. In the agglutination reaction test, the serum of the patient is added to a tube containing the pathogen that is suspected to cause the infection. Let's take Salmonella as an example. If the serum contains anti-Salmonella antibodies, those antibodies will bind to their specific epitopes on the membrane of Salmonella, and since each antibody has two binding sites, this will cause the agglutination of the bacteria and their sedimentation. And so, the appearance of sediments in the tube confirms that this person is infected with Salmonella. On the other hand, if the bacteria remain free in the tube and no sediments are observed, this indicates that the serum is devoid of anti-Salmonella antibodies and the patient is not infected with this bacterium. Document A of your books shows the difference between the positive agglutination test for a Salmonella-infected person and the negative test of another person not infected with Salmonella. Another technique used for infection diagnosis is immunodiffusion in gel. During this technique, the serum of the patient is placed inside a well in agar gel, facing another well that contains the antigen suspected to cause the infection. Both antigens and antibodies in the serum will diffuse in the gel. If the serum contains antibodies against this specific antigen, those antibodies will bind to the antigen and a precipitation arc will be formed. Document B of your books shows how this precipitation arc is formed only if the antibody is specific to this antigen, while it's not observed when the antibodies are not specific to the used antigen. The third technique to study in this document is ELISA, which stands for Enzyme-Linked Immunosorbent Assay, and is well known to be used in the diagnosis of HIV infection. During this technique, the antigen suspected to cause the infection is fixed at the bottom of very small wells. Let's take HIV antigens, for example. Then, the serum of the patient who is tested for HIV infection is added to this well. If the serum contains anti-HIV antibodies, those antibodies will bind to the HIV antigen, while other non-specific antibodies of the serum will remain free. Those free, unbound antibodies are washed out of the well, so that only HIV antibodies remain in the well. But if the patient's serum does not contain anti-HIV antibodies, then no antibodies will be fixed to the HIV antigens, and thus there will be no antibodies at all in the well after washing. Up to now, the results obtained in both cases are not visible to the lab technician, but the following steps of ELISA will give some visible colored results. At this step, enzyme-linked antibodies are added to the wells. Those antibodies can bind to the constant region of HIV antibodies, if found in the well, and they hold an enzyme at the same time. In this case, those enzyme-linked antibodies will be fixed in the first well, but obviously they won't be fixed in the second well that is devoid of antibodies in the first place. Now, wells are washed again to remove any unbound antibodies. The final step is to add a substrate, specific to the used enzyme. This substrate is transformed by the enzyme into a colored product. Thus, a change in color will only occur in the well that contains the enzyme. The change in color in the ELISA test is considered to be a positive result that confirms the presence of anti-HIV antibodies in the serum of the patient that will be diagnosed to be infected with HIV. Document C of your books shows the procedure of the ELISA test. The serum tested in this document belongs to a person infected with HIV, since a change of color was observed upon the addition of the substrate. The last technique to discuss in this document is immunofluorescence. What is different about this technique is that instead of searching for antibodies in the serum of a patient, antibodies are used by the lab technician to detect the presence of an antigen in a tissue. The antibodies used in this technique are fluorescent. That is, those antibodies are chemically bound to a fluorescent molecule that emits visible light when exposed to UV light. A sample of the tissue to be tested for infection is placed on a slide. Then, fluorescent antibodies are added. The slide is then washed to remove any unbound antibodies. If the tissue has the antigen, then antibodies will fix to it, and when the slide is exposed to UV light, those fluorescent antibodies will glow. But if the tissue is not infected with this particular antigen, no fluorescence will be observed. Document D of your books shows the green fluorescence observed upon testing a tissue by immunofluorescence. So by the end of this video, make sure that for every technique you are able to identify the following: the aim, the procedure, and the positive result. If you had any difficulty doing this, replay the video again. Hope you enjoyed the video. Remember to read the document and the related part of summing up, solve probing the document, and the following exercises.